residency in anatomic pathology at the WCVM. His MSc research focuses on American foul brood disease in honeybees. So let's welcome Dr. Ivanna Kozii with her first presentation.
Thank you for the kind introductions. And I think in veterinary community, we all know how insightful and useful histology can sometimes be, and how useful histologic tissue examination can be for detecting disease, diagnosing disease, and investigating an array of exogenous stimuli on the organism. So of course, in our field, we could not pass by an opportunity to review some of the normal microscopic anatomy of the honeybees during this seminar. So with this next 40 minutes, we will quickly review the exoskeleton of the honeybees and then move on to identify some of the structures found in the head and thorax of the honeybees, and then finish off with reviewing several organ systems that are located in the abdomen of the honeybees, both all three castes, the workers, queens, and the drones. And to begin with, we also, to spice the talk up a little bit, will include some of the function for certain aspects of the histologic structures that we'll be discussing. And of course, we will share some of the research that our lab does with the honeybees in the last couple of years. So to start with, we'll start with the exoskeleton. Now, the honeybees don't really have bones, but they do have an exoskeleton that is there to protect them from the environment around them. And their exoskeleton is composed of distinct plates that, if you look closely enough, you can even see how these plates are separated one from another. And two of the most important components of these plates of the honeybee exoskeleton are chitin and sclerotin, and when they work together, they make the exoskeleton both strong, but yet flexible, and also rigid and hard. And the exoskeleton composed of the chitin and sclerotin works wonders for the honeybees and protecting the honeybees from the environment around them. But unfortunately for us and for all of the histology enthusiasts out there, it also prevents us from obtaining really good quality histologic slides sometimes. And so these images here is what our lab started with 5 years ago, when we were really excited to delve into the world of honeybee histology. And I think with the next couple of slides, we'll prove that with a little bit of patience, a good microtome, and a graduate student with some time on their hand, you can always outsmart some of the tissue preparation techniques and figure your way around some of the difficulties that we face when starting new research projects. And I guess it's important to note that for our tissue preparations, we fix our honeybees in formalin. The tissues are then processed in a regulatory WCVM veterinary diagnostic laboratory, or Prairie Diagnostic Services, PDS. And we embed our tissues in paraffin and section them ourselves at 4 or 5 microns and stain them with regular hematoxylin using stains. And so all of the histologic images that will be shown in this presentation were obtained by a number of graduate students in our lab over the last 5 years. So let's start with the exoskeleton. Again, the exoskeleton has multiple layers, and the very top one is the epicuticle, which is this dark brown colored material that overlays the exoskeleton, and it is responsible for waterproofing all of the cavities of the honeybee. Right under the epicuticle is the procuticle that is composed of 2 layers, the exocuticle and the endocuticle. Now, these 2 layers are high in chitin, so it's a very strong and flexible material, but only the exocuticle contains high amounts of sclerotin, which is responsible for rigidity and hardness of the exoskeletal tissue of the honeybees. And similar to dermis, for example, it depends on which part of the bee you're examining. The thickness of these multiple layers is going to differ greatly. And so in this particular area, this is a section through the abdominal plate, and you will see that the exocuticle, which is this light brown material, is relatively thick, whereas the endocuticle is practically not visible. But luckily for us, there is another fold in this section here, was this eosinophilic, almost laminar material. So that's what the endocuticle would look like. And the last layer here is the epithelium that lies right underneath the endocuticle and is responsible for production of the exocuticle in bees. Some other things of note in the exocuticle are the mechanoreceptor hairs that are found all through the body of the honeybees. These mechanoreceptor hairs are highly innervated and help the bee orient and communicate with the outside world. And you will also see that the muscle in bees is connected directly to the cuticle itself. Another cool thing about the exocuticle and the distinct plates that the exocuticle is made up of is the way that these plates are connected between each other. And the connection is through these flexible membranes. And again, depending on where in the body you will look at them, the membranes will vary in size. And they will usually have multiple folds that allow for great expansion and movement of the plates between each other as the bee moves or needs to squeeze in through smaller spaces. So that's that for the exoskeleton. And we can move on to reviewing the histology of the honeybee head. So for orientation, in this image on the right here is just an image of the honeybee worker head. And you will see the two compound eyes on the right and left, and then one of the three ocelli or simple eyes sitting on top here, and the mandibles at the bottom. And on the left is a transverse section of the histologic section of the honeybee worker head. And similarly, we see the two compound eyes on the left and right, the start of two ocelli in the section, and the mandibles here at the bottom. And when we talk about histology of the honeybee head, there are really three main components that we would need to discuss, and they include the brain, some of the secretory glands, and the eyes. And for this presentation, we'll start with the eyes, the compound eyes specifically. In bees, a functional unit of a compound eye is called the ommatidia. And the way it works is the eye is covered with multiple distinct lenses, and each lens concentrates light onto a clear cell called the crystalline cone. Now, this crystalline cone conveys this light signal through a central core called the rhabdon. And the rhabdon is wrapped in retinula cells, which are these eosinophilic cells seen right around the central core rhabdon. Retinula cells are highly sensitive, and they are able to pick up the slightest light senses from the rhabdon and deliver them to the nervous tissues in the bottom. Now, to ensure that the light that is passed through a single ommatidia is not diffused and doesn't pollute the neighboring ommatidia, each ommatidia is also surrounded by pigmented cells, which are seen here in this amphiphilic color or light eosinophilic color. So these pigmented cells make sure that each ommatidia functions as a separate unit. And so with that, it's kind of easy to imagine what a bee vision looks like. So the picture that they receive in their head looks kind of like this image on the bottom left here, where each ommatidia produces a single dot of the big picture of the honeybee vision. And once the dots from each ommatidia come together, the bees are able to have a big picture of the environment around them. So the vision of bees is often compared to a mosaic or the pixelated image of a photo on a computer screen, where each pixel corresponds to a single ommatidia that has delivered a light that was passed through the sensors. Now, depending on which caste you look at, their compound eyes are of different sizes, and you will see that the workers and queens are somewhat similar or comparable, and they each have about 4,000 of these ommatidia in each of their compound eyes, whereas the drone eye is huge in comparison to the worker and queen. And that's because their eyes have up to 2 times more ommatidia compared to workers and queens. And that's not really surprising. As Amir mentioned, the sole purpose of a drone's life is to find a queen and mate with her. And because mating in honeybees happens mid-flight in the air, the drones require quite a bit of eye and movement coordination to catch up with the queen and be very targeted during their copulation. So that's one of the reasons why they have the bigger eyes and more ommatidia. Another type of eye that the bees have is a simple eye. Now, simple eyes are somewhat different. They only have one lens that concentrates light on a cluster of retinular cells. Again, these eosinophilic light-sensitive cells that deliver nerve impulses to the brain. And each retinular cell is also surrounded by these pigmented cells that prevent the spread of the signal, diffusion of the signal between the different cells. Now, the ocelli, or simple eyes, are known to be innervated with high caliber nerves, so they are able to deliver the nerve impulses really, really fast. And in terms of the function of these two eyes, they function somewhat differently, where compound eyes are responsible for the big picture image of the surrounding environment in bees. They are also responsible for detecting fast movement. So with the compound eyes, the bees are able to detect a change in their environment 10 times faster than a human eye would be. With the simple eye, however, they are able to direct their flight and sense the direction of the sun above them. So they need these eyes to work in cohesion for their fast flight and targeted approach to finding the flowers that they are after in the environment. So that's the eyes. Another big part of the organs that are found in the head are, of course, the brain. And in the honeybee brain, the mushroom body is probably the part that has been studied the most, as it is responsible for learning and memory. So as Alamir said, these bees that develop into bees that go out to collect nectar, they have to go really far away to find the flowers and then remember the way to the flower, remember the way back to the hive, and remember how to teach other bees inside the colony to go on the same commute daily or multiple times in a day. And so the mushroom body is where all of that learning and memory happens and is being stored. The mushroom body in honeybees is comprised of the two calyces, the medial callus and the lateral callus, the central core or the alpha lobe, and the beta lobe that is kind of wrapped around the alpha lobe. So that's the mushroom body. Other parts of the brain, of course, include the optic lobes that is composed of the lobula as well as medulla. There is also the subesophageal ganglion that is named so because it sits right under the esophagus, seen in this image as well. And these have quite well-developed antennal lobes because antennae is another sensory organ in honeybees that helps them communicate with the outside world as well. Now, honeybee brain is a highly metabolically active organ, and it requires loads of oxygen to function properly. And the way bees deliver oxygen is through an elaborate system of air sacs that are seen in this section. And these air sacs then split into tracheal tubules that are kind of seen here. And the tracheal tubules deliver the oxygen to the bee brain and help it sustain the functionality that it needs. The last thing that needs to be discussed in a honeybee head are the secretory glands of the honeybees. And these include the hypopharyngeal glands, which are these clusters of acini that have a single duct that empty into a common duct. The hypopharyngeal glands are responsible for brood food production. So these are the glands that produce food for honeybee larvae. There are also these thin-walled glands, which are the salivary glands that produce a number of enzymes that, when mixed with the nectar that bees collect, helps the bees transform it into honey that is stored in the hive for a long time. And of course, the mandibular glands. Now, mandibular glands have multiple functions. They are responsible for production of some pheromones, which translates into chemical communication in the hive, but they also participate in production of royal jelly, or the queen food or larval food in honeybees as well. And now some of these glands have been heavily investigated in our lab, and I will take this opportunity to just quickly review some of the findings that we were excited about and would like to share with the attendees. The first thing to note is that hypopharyngeal glands are very vary with the age and are highly sensitive to external stimuli. So for example, the size of these glands will depend on the age of the bee, where they will be the greatest in size in bees that are responsible for larval rearing, so larval feeding, which would be in bees between the first and second week of their life. We also know now that hypopharyngeal glands are sensitive to toxic stimuli and will decrease in size in response to some fungal and insecticidal stimulus or the combination of the two. Another important thing to note is that hypopharyngeal glands are also sensitive to infectious disease. One of the students in our lab, Dr. Ivanna Kozii, was investigating clinical signs of bees exposed to deformed wing virus in a natural environment. And in addition to observing how bees emerge from their cells and how they look grossly differently from their control bees, Roman also investigated the histologic appearance of the head of bees naturally infected with deformed wing virus and compared them to bees from the same colony that had no clinical signs. And so what he found is that the number of hypopharyngeal gland acini in control bees was much greater compared to the number of these acini in deformed wing virus infected bees. In addition to that, he also looked at the mandibular glands and found that mandibular glands in control bees were lined by plump, healthy epithelium, whereas the epithelium lining mandibular glands in infected bees was greatly attenuated. And so these histologic findings now let us know that the bees infected with deformed wing virus may not be the greatest at taking care of the brood inside the colony, in addition to some of the other clinical signs that have already been known. Also in our lab, we take great interest in investigating mandibular glands. But before I go there, I guess it's important to know that mandibular glands also vary greatly between the different castes. So you see that in a worker bee, the mandibular glands are fairly well developed, whereas in a drone, these glands are barely rudimentary and are even difficult to find on serial histologic sections. But comparing worker mandibular glands to the queen mandibular gland, the difference is quite drastic, with the queen mandibular glands being much greater in size. In addition, these glands in queens are surrounded by these fat cells, which are these plump basophilic cells seen around the gland. And fat body in honeybees is kind of like our liver. It is responsible for some detoxification, but also for some nutrient storage. So with queens, these mandibular glands are important because this is an organ where queens produce their pheromones. And pheromones in queens, just like Elamir said, are extremely important to keep the colony in cohesion. So in this picture on the left here, you see a honeybee queen that has been paint marked on the thorax. And you even see how her abdomen is bent inside a cell. So she's laying an egg. And all of these workers around the queen have come by to say hi to the queen and also to lick her, as funny as it sounds, because as they lick the queen, they get a dose of their pheromone that they are going to then go into the hive and share with the rest of the bees. And as long as this queen produces enough pheromone to keep all of the workers in the colony happy, the colony will thrive. However, if the queen does not produce pheromones or the pheromones do not satisfy the workers, the queen is doomed and she will be replaced either by the workers or the beekeeper who is not satisfied with the success and health of his honeybee colony. So with our research, we were interested to see if these glands are sensitive to insecticide exposure. And particularly, we work with neonicotinoids, which are neurotropic insecticides. And so what we did, we raised some queens from honeybee larvae. And when they were larvae, they were exposed to incremental doses of this neonicotinoid insecticide. And what we found is that queens that were exposed to 50 nanograms of thiamethoxam, or this insecticide, develop into adult queens with mandibular glands that are 26% times smaller compared to the control bees. Now, it is important to say that this dose is extremely high and is almost 10 times higher than any environmentally relevant dose. But as veterinarians, we also know that functional abnormality often precedes morphologic changes. And so this finding pushes us to think that, hey, if morphologic changes in mandibular glands are so drastic, maybe exploring and investigating functional abnormalities of the mandibular glands in queens is a useful tool for detecting toxicities. So this was one of the exciting discoveries that our lab has stumbled upon during our journey of honeybee research. But enough about that, and let's go back to histology and move on to the thorax. Now, the thorax in honeybees is basically a box full of muscle. So the muscles that are found in the honeybee thorax are longitudinal flight muscles, which are responsible for the downstroke, and then the vertical flight muscles, which are responsible for the upstroke. Other things that can be found in the thorax also include, well, the esophagus, which I'll have you note that the esophagus is highly folded, which allows for great expansion of the esophagus when the food is passing through. Some other things include the air sacs, which are seen here, which again is an elaborate system of honeybees to deliver oxygen to the organs and systems. Seen in this section also, there is a thoracic ganglion, which is part of the nervous system. In the thorax, there are also multiple breaks of the chitin, which are called the spiracles, which is the way through which the air is sucked into the body cavities of the honeybees from the outside world and then delivered through the body, again, through these air sacs. Now, the thoracic cavity also has salivary glands that didn't make it into this section, but are important to, again, produce honey from the nectar that bees collect. And moving on to the abdomen. Now, the abdomen houses several organ systems, and we'll start with the circulatory system. Unlike the mammals, the bees do not need blood to deliver their oxygen because they have their air sacs, but they do need hemolymph to deliver nutrients from one part of the body to another, but also to redistribute pressure between the different cavities of the body. And the way the circulatory system in honeybees is organized, they have a heart that spreads between 2nd and 7th plates of the abdomen. And the heart takes up hemolymph from the abdominal cavity and pumps it through the aorta into all of the body cavities. And as the hemolymph makes it into the head, it floats freely within the cavity, and through the function of other organs and compression and contraction of the muscles, it finds its way back into the abdominal cavity, where it is filtered and recirculated back into the heart. Histologically, this is what the heart looks like. So this is part of the heart, and you will see that the heart has a single, or actually it has multiple, but in this section, it has a single opening called the ostia. And that's the way how hemolymph finds its way into the heart from the abdominal cavity. And as the heart pumps, it uses the valves to ensure the unidirectional movement of the hemolymph from the heart into the aorta and again into the head. Another big part of the abdominal cavity is taken up by the digestive tract. Now, this is a busy slide, but stick with me. So the digestive tract of honeybees is composed of the pharynx in the head, and then the esophagus that goes through the thorax. And in the abdominal cavity, it starts with the crop. Now, the picture below is an digestive tract that was dissected carefully out of a single bee by one of our lab mates, Dr. Colby Klein. And you will see that the crop here corresponds to the crop here in the dissected section. Now, the crop in honeybees is kind of like a cargo container. So remember how Elimir said that bees are able to carry up to 30% in their weight of nectar? This is where it happens. They collect all of their nectar into the crop that has a great ability to expand and take up most of the abdominal cavity in worker bees. And as they transport the nectar in the crop to the hive, they need to make sure that this nectar is not going to go further down the digestive tract. And the way they do that is through their second mouth, or proventriculus. And the way proventriculus works is it has 4 lips that are able to reach into the crop and filter out pollen bits, which is the bee's protein source, from the nectar within the crop. And that's how they take up pollen into the ventriculus, which is the true stomach and the only part of the digestive system that is not covered by chitin. Now, from the ventriculus, the digestive tract continues into intestine and the rectum. Histologically, this is what it looks like. So this is the crop, remember, the one that stores the nectar and is able to transport the loads of nectar from the flower into the colony. And here is the proventriculus. And I feel like you can even imagine how this proventriculus, like a clam, can go into the crop and sieve out the pollen bits and take it all the way into ventriculus. You can see that this little bit of proventriculus is called the proventricular sleeve, goes into the ventriculus itself, and prevents retrograde movement of digesta from ventriculus into the crop. Now, looking more closely at it, this is more of a cross-section through the ventriculus. You can see, again, the forelips of the proventriculus and the highly undulating lining of the crop that, again, is undulating because it needs to allow for great expansion. Now, from proventriculus, we will move on to the ventriculus, which again is the true stomach of the honeybees. And it is lined by ventricular cells that produce this peritrophic membrane. Now, peritrophic membrane serves two purposes. First, it mixes the digesta with the enzymes and therefore stimulates the digestion of the food that bees take up. But also, it protects the ventricular lining from any physical and mechanical damage that bees can take from the pollen. Ventriculus is an important organ to investigate in some bacterial diseases in honeybees. So for example, these two images are images of the larval ventriculus, where the ventricular lining is these basophilic cells on the left, and the blue stippled material inside is Melissococcus plutoneus, which is a causative agent of European foul brood disease. And you will see that the larva on the left has low infection of this disease, and the bacteria just kind of sit on top of the peritrophic membrane and that separates the bacteria from the ventricular cells. Whereas on the image on the right, the bacterial overgrowth is so great that they have practically consumed all of the peritrophic membrane that separates the bacteria from the ventricular lining. And the larva at this point dies really from malnutrition, which is just kind of cool to know. Now the ventriculus goes on into the intestine. And again, on this image, just for review, we see this proventriculus that reaches its mouth pads into now expanded crop in this bee. And then it goes into the ventriculus. But what's more cool in this image is that we can see the transition between ventriculus into the intestine. And it just so happens that this transition is exactly where the Malpighian tubules of the honeybee open up into the digestive. Now, Malpighian tubules in bees work kind of like our kidneys. They filter out hemolymph, and all of the junk that they filter out is then released into the intestine. And the intestine will then include the intestine itself and the rectum. The cool part about the rectum is that it is able to expand by great volume and hold the digesta of the bees for several months on end during the winter, at which time bees are not able to go outside of the colony and empty their rectum because the flying conditions aren't always great. But the rectum also serves another purpose. Unlike the intestine, the rectum has multiple breaks of epithelial lining that allow reabsorption of water from the rectum into the hemolymph. So that is it for the digestive tract. And we can move on to the reproductive system, which I'm slightly biased towards, as I find this one to be my absolute favorite, at least in honeybees anyway. So for honeybee queens and drones, similar to other animals, they have their gonads where the gametes develop. So the queens will have their ovaries where development of eggs takes place, and then the tubular part of the reproductive system through which the eggs travel. And unique to honeybees is their Spermatheca that Alamir called the nitrogen tank, remember? So the Spermatheca where the queens collect the sperm and are able to keep it viable for 3-4 years as they live inside the colony. In honeybee drones, again, similarly, they have their gonads. So they have the testes where sperm development takes place, and they have the tubular part of their system, which includes the vas deferens and the seminal vesicles and the endophilus, which is their sex organ. And they have some sex glands, specifically the mucus gland, which produces specific mucus that comes in handy during copulation. But unique to drones is that the morphology of their reproductive system is highly dependent on the age at which you will look at the drones. Just for some background information, if you think about the size of testes, we are used to thinking that in most animals, as they mature through the adolescence, the size of their testes will increase, like we see in bulls and dogs or humans, for example. Now, we also know there are other animals that are somewhat different, where the size of their testes will increase as they reach sexual maturity, but it will fluctuate with the as the mating seasons come and go, like we see in deer and moose. However, in drones, it's completely different because their sperm development or spermatogenesis takes place as the drones are still in their pupal stages. And so when the drones emerge or are born, their testes are of the greatest size. And as the drones mature, the testes decrease in size because the sperm moves from the testes into the tubular part of their system. And therefore, one of the projects that Dr. Colby Klein did in our study, in our group, he carefully dissected the reproductive system of drones at different ages of their development. So from day 1, 2, 3 to day 15. And what you will notice is that the size of the testes changes drastically from the time the drone is born onto the time that the drone is considered sexually mature at day 15. And the reason for that is that the sperm that is mature will move from the testes into the seminal vesicles, where it will wait for its turn to be used during mating. So if we look at the histology of the drone reproductive tract, this is what we would see. This is a longitudinal section across the abdomen of the honeybee drone, and you will see that the majority of the abdomen would be taken up by the testes at this stage on day 1 of after emergence or after birth in drones. Now the sperm from the testes will then travel through vas deferens and into the seminal vesicle, where it will be stored until further use. And caught in this section too, we see the mucus gland. Again, and the secretion of the mucus glands will be used during copulation. And if we were to take a closer look at a single testicular tubule of the honeybee drone, this is what we would see. So this is the outline of a single testicular tubule. And I know that specific cells are kind of difficult to see in this section, but to help you imagine these cells, there is this dotted line. So the black dotted line signifies the follicular cells. Now, follicular cells is the place where sperm development takes place. So each follicular cell will house multiple follicular cysts. And within each follicular cyst, a cluster of sperm will be developing. And depending on which stage of development the sperm is at, they will get different names. So for example, this when the sperm has large clustered nucleus, it would be called the spermatids. And when the sperm starts to look more conventionally to what a developed sperm would look in other animals, we would call them spermatozoa. Now, when these cluster of sperm will develop, it will be expelled into the lumen of the testicular tubule, and a single cluster of the sperm will be called the spermatodesms. If we were to look at the same testicular tubule, but further down the line in a longitudinal section, this is what we would see. So remember these clusters of sperm, these are the spermatodesms that are making their way from the testes into the seminal vesicles. But also in this image, we can still see the follicular cells that are still quite plump and still have those follicular cysts with a cluster of sperm that are in a similar stage of their development. Now, if we were to move from day 1 to day 10, you can already see that the size of the testes decreases almost by half. And if you look carefully on histology, this is a longitudinal section of the same testicular tubule, and you compare it to day 1, where it's full of spermatodesms. At day 10, it's already pretty empty with only rare spermatozoa seen here and there. And you will also notice that the follicular cells are much less are deflated and have much less happening inside their cell. For a general conclusion, when you look at drone reproductive histology, it will depend on the age at which you're examining the drones. And again, on day one, the testes will be the greatest and the tubular part of the reproductive tract will be empty. But as the drone matures, the testes will decrease in size and atrophy greatly, while all of the sperm will move on from the testes into vas deferens first, and then be stored in the seminal vesicle until the drone is copulating, at which time the sperm will be expelled through the endophilus into the queen, into her spermatheca, which we'll talk about later. Now onto the female reproductive system. So with the queens, their eggs are developing inside the ovary. And a single oocyte or an egg, developing egg, and then a cluster of nurse cells or trophocytes that are used to support the oocyte throughout its development. And as the egg matures and moves from the apical portion downward, you will see that the size of the egg increases, and the eggs at the bottom of the ovary have no nurse cells clustered around them. And that's a clear sign that the egg is mature and is ready to be laid. So in this picture, you can already see the egg that's mature and is getting really close to the tubular part of the reproductive canal. And the reproductive canal starts with the calyx, which really is like a funnel structure that hatches the egg and directs it from the ovary into the tubular portion, which is the lateral oviducts, seen on this histologic image here on the left and the right. Now, the cool part about lateral oviducts is that the lumen, inside the lumen, there are these chitinous septaniform spines that promote unidirectional movement of the egg from the ovary into downwards the reproductive canal. Now, the lateral oviducts will come together to form a median oviduct, which is a highly mascularized structure through which the egg will pass into the bursa copulatrix and will be laid. Now, the cool thing about median oviduct is that it has a specific structure called the valve fold, which is a muscular flap that is able to practically close up the median oviduct as the egg passes through. And the reason for the valve fold to be there is because it sits right underneath the spermatheca. So when the egg is laying an egg, when the queen is laying an egg, the egg needs to be fertilized with a sperm that comes from spermatheca. To ensure that the egg is not going to escape and slip through the reproductive canal too fast, the valve fold slows down the progression of the egg through the median oviduct so as to give a chance to the sperm to catch up with the egg and to be fertilized. Now, the cool thing about these here is that unfertilized eggs that are not fertilized with the sperm will develop into drone bees or male bees that are haploid. And when the egg is fertilized with the sperm, the bee will develop into a female, so the worker or a queen. And the way that the queen decides whether the egg is going to be fertilized or not fertilized depends on the size of the comb that the egg is going to be laid in. So the queen, she will carefully inspect the cell before laying an egg. She will go in there with her legs and her antennae, and she will measure the distance of the cell. And if the cell is approximately 5 millimeters long, it will lay a fertilized egg that will then develop into a female bee. However, if the cell is slightly larger, and you can already see that drones are quite a bit larger compared to workers. So if the cell is larger and is around 5.5 millimeters, the queen will opt to lay a non-fertilized egg. So that's the way the castes between females and males are decided. But between the worker and a queen, this fertilized egg, there's no difference. So any fertilized egg has a potential to develop into a worker or a queen. And the only difference that decides which type of bee will develop from that egg is the type of diet that the bees are provided with. So the queens are fed only royal jelly throughout their development. And remember the royal jelly that is produced by the hypopharyngeal glands of the worker bees and the mandibular glands of the worker bees. Whereas the worker bees, throughout their development, they will be started off on royal jelly, so the production of hypopharyngeal glands and mandibular glands, but later on during their larval development, the nurse bees will mix in a little bit of pollen and a little bit of honey into their diet, which will prevent the worker bees from developing a fully functional reproductive system. So between the two castes, the workers and the queens, there are some distinct differences in their reproductive system. So the worker bees are thought to not have active reproductive system compared to queens that have about 300 ovarioles, which are these thin threads through which the developing egg travels. The queens have about 300, whereas the workers only have about 3 to 12 ovarioles, which is significantly less than the queens. In addition, some of the stinging apparatus is also going to be different between the worker and the queen. The worker stinger is going to be barbed, so really it's a single-use tool, and the minute they use it, they get stuck in it, and to be let free, part of their digestive tract is going to be torn out of their abdomen, which is why they die after they sting you. Whereas for a queen, that's different because their stinger is smooth. And although they don't really use it for protection of the hive, they do use it when they are fighting other queens within the colony in early stages of their life. But the most important difference between the worker and the queen is the presence of this Spermatheca in a honeybee queen. Because the workers do not have a Spermatheca, they're not able to mate, and therefore they would not be able to produce female offspring, even though they can lay eggs because they do have ovarioles. Now, for queens, the Spermatheca is extremely important because, again, it's the nitrogen tank. And so this is where the queens will collect all of their sperm that is then being used up throughout their development. Histologically, this is what it looks like, with the Spermatheca being lined by a single layer of epithelium. And this is a Spermatheca from a mated queen. And you can see how tightly clustered the spermatozoa are within the Spermatheca. It's also equipped with a spermathecal gland that provides the sperm with nutrients and some other useful materials during the life of a bee. And here in section, we can already see the spermathecal duct through which the sperm will travel into the median oviduct. Now, I know that majority of this presentation could have been presented probably in this single slide with the honeybee head and that shows the simple eyes and the compound eyes and the brain of the bee and the secretory glands, the thorax with the musculature of the bees, and a section of the abdominal cavity. But I guess in veterinary medicine, we are too excited about histology to make a presentation that would be worth 5 minutes into 40. So with that, I would like to also thank the sponsors that make our research possible here at WCVM. And I'm not sure if I have any time for questions. Oh, Q&A. Do I have time for questions?
Yeah.
Okay. Do they treat with something to soften the exoskeleton prior to fixing or embedding? I wish we knew a good chemical that would do that. We were unable to find one that worked really well for us, but we do sometimes dissect part of the exoskeleton away so that it's not on the way of the knife as we section our slides on the microtome. You stain differently. Maybe I missed it. Are you staining differently to highlight cuticle? Thank you. No, so all of the stains done in our lab are regular hematoxylin and eosin staining. Is that separation between the lens and the crystalline cone on the histology slide artifact, or do they have something like the aqueous humor between the two? My understanding, the separation on our histologic slide is artificial. My understanding is that they don't have the aqueous humor like we do, but I would double check on that. Brood food glands is like mammary glands. The food is secreted by them. Do the workers eat pollen, honey, bee bread, and then secrete food for brood exclusively? So the way it works in worker bees, they eat a lot of pollen, which is the source of protein. And then they use this protein that they get from pollen to produce secretions from the hypopharyngeal and mandibular glands. And again, for queens, the secretions from these glands is the only source of food that they will get throughout their life. Whereas for workers, a little bit of pollen and honey will be mixed in, or bee bread will be mixed in with their food. Can you explain more about how the hypopharyngeal glands produce brood food? Is that royal jelly? So I'm not quite sure on the mechanism of how hypopharyngeal glands produce food, but I will say that the secretion from hypopharyngeal glands and mandibular glands together, that's what makes up the royal jelly. How do you measure size of mandibular glands or other organs in bees? Is there a particular imaging program that you would recommend? There's an email, and we can definitely send you an email for that. But in our lab, we do serial sections of the head in this case, and then we evaluate the serial sections and take total area of organs of interest. So when you can see their abdomen pulsating, it's just their heartbeat. I don't know, but my understanding is that the heartbeat would not be as strong so as to make their entire body pulsate, but they do have other muscles within the abdomen that would be stronger, I guess. They may have missed these points. Do you fix in 10% formalin or something else? We do use 10% formalin. That being said, other research groups that publish with bee histology can use other fixatives as well. Do you have any specific fixation or processing instructions for histology to step out for me? Yeah, so the paper that was referenced by Dr. Ivanna Kozii in VetPath has a quick review of how we prepared those slides, and that could be a good resource.